Okay, as the famous poet Jimmy Buffett said, the clock says now. So let's go ahead and get started. So good afternoon, everybody. Welcome to the 63rd Faculty Lectureship at NDSU. My name is Tom DeSutter, and on behalf of the Faculty Awards and Recognition Committee, I would like to welcome the relatives and friends of Dr. Achintya Bezbaruah and all the members of the community, President Cook, Provost Bertolini, faculty, staff, students, and everyone joining online. This year, the awards committee has recognized Dr. Achintya as a recipient of the lectureship, one of the most prestigious awards at NDSU. This afternoon, we would also like to recognize Dr. Gary Secor, who was a finalist for this award. He is a professor of plant pathology. The faculty lectureship recognizes sustained professional excellence in 3 areas of teaching, scholarship and creative activity, and service. Dr. Bezbaruah has achieved impressive accomplishments in each of these, and you will hear more about them in a few moments. Dr. Bezbaruah is the most recent of the 51 recipients of NDSU's faculty lectureships dating back to 1957, with professors speaking on a range of topics, including Arctic exploration, the conductor's art of ice and men, those of you who would kind of clever, 2 years on a pandemic frontline. And one of my best friends gave a lecture, Dr. Jay Leach, in 1996, Wetlands, Why the Controversy Continues. And so following Dr. Bezbaruah's talk, there will be time for questions. Those joining us remotely will be able to submit questions online at that time. And Provost Bertolini will then present the Faculty Lectureship Award. Following the ceremony, we hope you can join us in the Butte Lounge for some cake and refreshments. I will now turn it over to Dr. Alan Kallmeyer, the Interim Dean of College of Engineering, who will introduce this year's Faculty Lecture Awardee. Thank you.
Thank you, Tom. Good afternoon, everybody. My name's Alan Kallmeyer. I'm Interim Dean for the College of Engineering. And it gives me great pleasure to introduce my friend and colleague as this year's recipient of the Faculty Lectureship Award, Dr. Achintya Bezbaruah. Dr. Bezbaruah is a Gehrts Presidential Professor of Environmental Engineering and currently serves as interim chair of the Department of Civil, Construction, and Environmental Engineering. He joined NDSU as a faculty member in 2005 after receiving a PhD degree in civil engineering from the University of Nebraska-Lincoln. Dr. Bezbaruah's research focuses on the food-environment-water nexus, specifically drinking water and plant nutrition. As a Fulbright scholar and internationally known scientist, he has promoted collaborative environmental research and education to address global challenges, believing in the power of collective efforts to alleviate them. He also serves as the director of the NDSU Grand Challenges Scholars Program, a comprehensive research immersive program for energetic and ambitious undergraduate students. He is truly a dedicated and distinguished scholar and educator, and I am proud to call him my friend. Please join me in welcoming Dr. Bezbaruah to the podium.
Good afternoon, everybody. Thank you for joining here today. It is such an honor to be presenting this year's faculty lectureship, and really an honor. I thank the committee, award committee, and thank Tom and Elizabeth for being here today. I also thank Kalpana and Eugene for nominating me for the award, and I'll specifically thank Anne and the Provost Office for coordinating all the effort. And this particular graphics is designed by University Publicity Department. So thanks, they did a very nice job. And thanks to the Provost Office for facilitating this particular function today. And thank you all of you for joining and joining online. My family is here. My extended family is here in Fargo. We got an extended family of quite a number of families. And my neighbors are here to see you, all of them. And good to see all of you, and my colleagues and my students are here. So let's get started on the presentation. So today's presentation will be, as you can see, from rainfall to snowfall. I'll explain that, why I named it that way. So I'm originally from a place called Assam. Assam is known for tea. If you have not taken tea, Assam tea is very famous. And also it is known for one-horned rhinoceros. So typically you see a two-horned rhinoceros, there's an African one. One-horned rhinoceros is very famous. I belong to that state. And interestingly, that state gets a lot of rain out there, a lot of rain. As you can see, Fargo gets about 13 to 20 inches of rain, and Seattle gets the most so far, 50 inches, and Assam gets about 200 inches. And highest recorded rainfall is 1,000 inches of rainfall. And not exactly in Assam, but in a close by place in Assam. So Assam, actually, that river that flows through it, about a 2 mile wide. And it's a red river also. It's called Red River. So that really placed me very well, being in another state, which is a red river. Only thing I suppose we're missing, my state, home state, where I was born, Assam is known as a land of red river and blue hills. And there's no blue hills out here. When I came for interview, Ikalak Khan was the chair. I came from Omaha to interview. Omaha, as you know, a little bit of undulation is there. Not exactly hills, but they're undulation. So I asked Ikalak, is there a hill here? Oh, he said, I'll show you one. So he took me to 7th Avenue and 45th Street. And he showed me the dumping site. And I believed in him that this is really a good hill. But, I think believing in him doesn't reflect on me very well, being an environmental engineer. I should have known there's a dumping site. I failed at that point. But anyway, I got the job at that point. So now, why I'm telling you this story, because it's very important for us to make connections. You do not work for money. You work for a passion. You work for something to achieve. So making a connection with the place you make your new home is very important. So I was looking for connections. So before I get into there, as I said, I'm originally from Assam, a place called Bokakhat. So my father was a entrepreneur, and we had like original petrol buses, we used to run. And when diesel engine was discovered, my dad's business plummeted like anything. But he recovered from there. And we have a lot of land holdings out there. And we are a big family. We're 7 brothers and sisters. And all of us are one way or the other teachers. So that's a good part. We all understand each other's, we empathize with each other's, all poor teachers out there. Anyway, so we have been very fortunate that we have been able to sell our land holdings. And we have been able to establish a science college out there about 30 years ago. And this science college is a unique one because it is a 100-mile stretch. There's no science college earlier. So this is unique, and science college is really, really doing well. This is named after my dad. So this is my ancestral home. This is the last property we had after we sold all our land. And we have donated this property to a nonprofit called Vivekananda Kendra for women empowerment mostly, and also agriculture innovation. So that is what we did. And we are very fortunate that, most fortunate part is not that we have been able to donate, but 7 of us being agreeing to do the same thing. I think that is the amazing part about it. So now I'm from an agricultural family. So in our house, in our family, we used to have a lot of land holding, and rice was the main crop we used to do. So we used to do rice, and it is something, very proud of. I still connected myself with rice. I wanted to be an agricultural scientist. I do not know really, not very sure why I become a civil engineer, So I become a civil engineer anyway. So then I was trying to connect back. So, when my elder brother, he's 3 years older to me, he will tell me that, okay, let us cut this particular taro leaves. We call it kosu, taro leaves. We cut them and we cart them to the field. And we also collect another thing, vitex. So that particular variety of weed, we collect them also, along with cow manure. We cart them all and put them in the agricultural field without knowing why you're doing it. But we did it anyway. Everybody said it's good for your agriculture. We did it. I think that's my connection with plants. I really love plants. So I want to continue with that. So I think this is something very amazing, because if you can produce a lot of food, you can feed a lot of people. So again, I think that's the connection I was trying to make. I'm here more than 7,000 miles away, and making a connection is very important, as I told you earlier. So I have to see the purpose of I being here. So then we started, okay, this, when I joined here, we started, on a project, some project. We did a lot of other stuff along with that. We worked on an interesting project for phosphorus. So phosphorus actually is a very interesting product. As you know, that plants need NPK, nitrogen, potassium, and phosphorus. So while nitrogen is abundant in the atmosphere, even though process is very dirty to produce, but you still have a lot of nitrogen. Potassium and phosphorus are the two which are like petroleum. They are non-renewable. So we have to now, potassium is still there, but phosphorus can get exhausted anywhere from 50 years to 200 years, depending upon how we spend it, how we use it. So as you can see, in 2033, so we have more demand than production of phosphorus. That's a major concern. So if you look at US, we are really in a vulnerable position because we have no reserve for phosphorus at all. So we thought, why cannot we think of some other sources? Because phosphorus is mined. Can we mine phosphorus from some other sources? So that is where we went. If you look at eutrophic lakes, you see a lot of algal growth. That is where we have our resources. Can we mine them? Mining them is very difficult because the concentration out here is so less. They're in microgram per liter. So 30, 40, 50 microgram per liter. So it's very, very less. But still it is a resource, So we started working on that one. So talking about, we thought we'll use nanotechnology to achieve that. Most of you what know nanotechnology is, nano is one level lower than, 3 level lower than micro. We know microscope and microorganism. So nano is 10 to the power minus 9, very small particles, very, very small. Our fingernails grow about a nanometer every second. That is the level. And we can produce nanomaterials like that. And after COVID, all of you know that we use nanoparticles very efficiently. Nanoparticles are nothing new about it. So we started working on iron-based nanomaterial. Why iron-based? Because iron happens to be the most benign and most abundantly available material, and we can produce that very easily. So we started working with iron nanomaterial, and we used them for growing spinach. So not exactly right away, we didn't use the nanomaterial. What we did is we used the nanomaterial to absorb phosphorus from eutrophic lakes. We simulated the condition. Of course, we also did a real sample. So we could absorb phosphate from eutrophic lakes. And then we thought, if we have absorbed phosphorus, why cannot we reuse it? That is the whole purpose of doing it, right? That is why we are doing it, because we want to create a circular economy. So we thought, why cannot we use it? So we used basically the nanomaterial that has absorbed phosphorus to grow spinach. And they grew very, very well. They grew very, very well. And what we have found that if you use the, I'm just trying to hear it, yeah. The spent one, spent means it is absorbing phosphorus. So if you use that, you can see that, profuse growth actually, very nice growth out there. So that's working very, very well. And as we analyze more, one of the things we have found, yes, it is providing phosphorus. It is also providing iron along with that one. And that's the amazing part about it, because if we can really fortify spinach with iron as well, we may be able to achieve something more than we thought we'll be able to achieve, because anemia is a major problem world over. More particularly, pregnant women has to take supplement to overcome the deficiency, iron deficiency problem. So what we did is, in a typical spinach that you buy from store, iron content is about 4%. Iron content is 4%. What we have been able to do, we have been able to boost up the iron content about 8%. You know, this is amazing. And this guy out there, you see, and as you know, that was a mistake. Do you know that? That whole cartoon was a mistake because somebody in Europe wrongly predicted the iron content in spinach to be 16%. And some cartoonist thought it's a bright idea. Why don't you create that, muscle man with iron taking in there? So that was the cartoon created sometime back. That was not the real one. So with our research, now the cartoon will be real one because we are able to fortify it a lot, right? So now it was so successful. We actually got a cover page in the Sustainable Chemistry and Engineering Journal. So that was amazing because nobody has done that work before because the way we did it. Now, can we eat the spinach? We don't know. That is something altogether we have to do altogether. We are still looking for volunteers, maybe, if you want to join. So this is something very amazing because this really gave us a kind of a shot in our arm to continue doing further research. Now, coming back to what we did now as we are doing research, I was trying to find the connection. This is very important, as I told you. Now, as we are starting to look into these things, what exactly we did when we were a child, now it is very obvious because iron, is very much there in taro leaves, and that Vitex we use, that is an insecticide, and cow manure that we all use in our yard, that contains phosphorus, nitrogen, and a lot of carbon. So we didn't know that, we used it. Now we are actually starting some work in our lab right now to understand how this compound bind together and how does it help in retaining, manure in the system. So that really helped us, motivated me to continue doing further work. So now, when I'm saying me, I know my students are sitting out there, they may be thinking, we did the work, you were claiming it to be, no. I mean, when I say I, it is we, remember that, okay? So, I just taking the credit. Normally, most of the presentation, my students are not there, I claim it to be, I have done it, now looks like I have to tell, you have done it, okay? Now, so we wanted to work because that work, iron nanoparticle worked very, very well. So we thought, why not we try some additional, sustainable material, sustainable material. So when we talk about sustainable material, circularity is very important. That means from the cradle to all the way to the grave, it should have no problem, no issues. It should be benign in nature. So we should look at everything. So we started working on this one. So we looked into some of the iron impregnated chitosan bits. Chitosan is one of the very interesting, polymers, which have been working for membrane development and others. So this is one of the work Umair has done. So he's doing right now. So we have been able to actually absorb phosphorus very effectively from water that contains phosphorus. So as you know, phosphorus actually not only in eutrophic lake, also in wastewater, you have a lot of phosphorus. Wastewater has actually very high concentration. That means that comes from your toilet and everything that contains. Also urine contains a lot of phosphorus actually. So we may be able to recover those. This is working very well. And also, earlier we worked on some of the montmorillonite clay and iron cross-linked, alginate bits. That one also worked very, very well. Tonoy worked on that work and it worked very, very well. Right now, Anand is working on some of the material where we are using, some of the agricultural waste and we're converting them to cellulose, and we are converting them again to activated cellulose, and we are functionalizing them. We are functionalizing them to absorb phosphorus. And once we can remove phosphorus, then you can use it for other purposes. Other challenging part is how to remove nitrogen. That can be another altogether different domain, but that is also our purview. So this, once we can absorb them, that we'll be doing a lot of desorption studies and others, so we'll be able to also supply them to the plants. Now, this is one of the interesting work we are also working. This is, Sirso is working on this one. This is the NIFA supported project. So this is an interesting project. Most of the time you all know that when you write a proposal, you write a lot of things, then, sometimes you anticipate that's going to happen, right? Then of course you do the work, right? When you get the grant, you said, okay, we got a grant, what to do now, right? So now this is exactly, sometimes, even though in this proposal, we wrote whatever we wrote, without any hiccup, we have been able to do the work, whatever you wrote in the proposal. So this is Mohi and myself, we are from NDSU. This is a joint project with Connecticut and University of Central Florida. So what we have been doing, typically when you use phosphorus, plant use efficiency of phosphorus is only 17%. 83% of the phosphorus that you use to field go waste. So can we increase phosphorus use efficiency of plant by 20%? 20% is the kind of a mark that we can call an advancement. We have to achieve that. Now with phosphorus, US soil, US has two types of soil: alkaline soil that we have here, and on the other side, a little bit on the eastern and the western side, we have acidic soil. Now in the alkaline soil, the phosphorus that we use doesn't melt at all. So efficiency is even low. Now we thought, can we create a micro nano environment where our phosphate fertilizer will melt easily, and we can do a slow release. So what we did a layer by layer, technique. So we put a layer of chitosan. Again, chitosan is very important. We are trying to overuse possibly chitosan. It's such an amazing polymer. Then we put an anionic polymer, multi-branch anionic polymer, because this acidic polymer basically. So that acid is going to go into the rock phosphate and going to melt it down. And chitosan is going to prevent quick, coming out of the phosphorus. So now we put another layer and put another layer. It's called layer by layer technique. So we want to try out how many layers we need to do. So this is very interesting work. You're thinking, oh, you're doing it. Actually, it's such a good, interesting project because it's the perfect marriage between civil engineering and agriculture. Because why? Because the whole process is a chemistry, basically chemistry process and agriculture unit. So in civil engineering, we use what is known as a fluidized bed. So we'll be using a fluidized bed to mass produce this one. And we actually applied for patent. We are very positive of this one, that this one will be successful because it's very unique. Nobody has done it before. Now, how it works, because your rock phosphate is, you're negatively charged, then we put a chitosan, which is positively charged. It's a very simple process, very simple process in theory, but complicated to implement. But it is just a layer by layer technique based on charge. So we have been able to do it very successfully. So now what we have found based on our research, we have been able to convert the rock phosphate to nano size. And we got a very good distribution of size here, about 110 nanometer, which is slightly above conventional nano size up to 100 nanometer. Then also what we got is we got very good, zeta potential. What's the importance of zeta potential? They're not going to agglomerate together. That's very important. They're not going to agglomerate together. So basically, when you spray them, so they will be individual particles going into the field. I think that's very important. And those are basically confirmation. And the last slide is just a confirmation of that we are really being able to put coatings on the rock phosphate. Now, what we have found is that we have done some, because the important thing is how phosphate is going to come out to the plant. That's the most important part. What we have found initial, first few days, there's a huge flux of phosphorus coming out. And then it levels down and it continues for, we are continuing this study for more. It's about 2-month study we have completed. So this is really, our target is it should be able to supply phosphorus for 3 months at least continuously. So that will be good because plants need initial growth stage, they need a lot of phosphorus, and towards the end also they need a lot of phosphorus. So we have been able to do that. One of the requirements for slow release phosphorus is that this release, initial release, should not be more than 15% of the content of total phosphorus. So we have been able to achieve that. So we are kind of admitting that, US doesn't have a kind of strict regulation, but European Commission has one. So we are following European Commission's guideline to develop this particular phosphorus. So this so far showing us very good result. We are still optimizing the layers, how we can do that. So this is something, because more layer you put, more expensive it is going to be. We are trying to optimize the whole process in the thing. Now, so that puts us in a situation, now we can call our research to be agricultural nanotechnology. We are going step by step, not really claiming that we are doing this. We are really doing something which is agricultural nanotechnology. So let's go into their agricultural nanotechnology part. Now, I like draw your attention to 104 and 107. 107 is the one we got lettuce grown in normal solution, normal, fertilizer solution. And this one, this one has some nanomaterial added to that one. And we saw profuse growth in there. We saw a lot of biomass in there. Lettuce, you eat it for the biomass. Lettuce doesn't have anything, If you're planning to have lettuce tonight, so to disappoint you, lettuce is all water and fiber. That's it. You don't have a lot of nutrients, right? Unless we try to fortify them, okay? Now, so this is amazing because you got huge biomass, right? Then Robert Brueggemann, who used to be a plant pathologist, he's a good friend of mine. He came to my lab. Oh, Achintya, your work is going very well. I told him, very proudly I showed him that my plants are growing so tall with nanomaterial. He doesn't look very impressed. He told me, Achintya, you have a son? I said, yes. When he was 2 years old, he was 5 feet tall. Will you be worried? I said, yeah, I'll be worried. So what you will do? He said, I'll take him to a doctor. I said, that is exactly what you need to do for the plants. Plants need to go to a doctor. The doctor was, of course, Bob promoted himself. He wanted to be the doctor. So what we thought, because I didn't understand this initially. Then we sat down for a month. We discussed about genetic part of everything. Then you realize there must be immune compromise in this material. So there is immune compromise. So Bob is the perfect person to work on that one. So we wrote without doing any experiment, we wrote an NSF project. We got funded. Because it's such an interesting idea to look into plants' innate defense mechanism in the presence of nanomaterial. So we started that work, So now, of course, this is not normal. So what we did is, we also, prior to that, we did another grant from USDA NIFA to look into similar thing, not exactly a defense mechanism. But here, what we have found, that if you introduce nanomaterial to a plant, a lot of those genes are either upregulated or downregulated. A lot of those genes, That's very normal, because if, our genes also really react to something, some fear or something. Many times we react differently. That reaction is always there. And we can capture them at 0 hour, 6 hour, 24 hour at different times. And this is one of the snapshot, at 6 hours, what are the genes were upregulated and downregulated. Then of course we thought, oh, let us dig deeper into that one. So because when you sequence something, you have millions of data, millions of data. So you need to find out what exactly is the cause of this one. So now, when you looked into this one of the genes, MAPK genes come out to be, very either upregulated or downregulated markedly. So MAPK genes are the defense genes. So they are responsible for the series of them. They're responsible for plants' defense. So that is the clue for us. This is a clue for us. Okay, there's something happening. So then what we did, we decided to do another experiment. So what we did, Bob was a barley person. So we took a resistant variety of barley. As you can see, resistant variety of barley. This is a resistant variety. And we introduced a necrotropic bacteria to that one. So, ideally, when you introduce necrotropic bacteria to a resistant variety, there should not be any disease at all, because it's a resistant variety. Now, what we did in this experiment for the first time, a lot of people work on plants with everything, but what we did, we thought of doing slightly differently. We call it double interaction. So what we did is, we introduced nanomaterial as well as the bacteria together for the first time. So when we did that, what we found that the defense really crumbled. The disease really propagated in the barley variety. So it became diseased kind of it. So it shouldn't have happened. Shouldn't have happened. So we kind of looked into that one. We looked into, this is only with nanomaterial, this is with, nanoparticle plus the pathogen. This is only with the pathogen. When you look into this one, so you see that even after 48 hours, that gene upregulation and downregulation really remained high when we do what is known as a double interaction, nanomaterial and pathogens together. That happened. So that's a serious concern. That means if you use zinc fertilizer in your field, which is nano, and you are doing barley, and you have disease coming to your field, your barley will be wiped out. That's exactly what it is. What does it mean? Okay. So now, of course, is it a failure? No, is it not a failure? Because plants still need zinc. So we have to now modify our nanomaterial, maybe put a polymer coating or whatever. So we want to modify that nanomaterial to get a better nanomaterial such that plant can still take zinc or whatever nanomaterial we give. We tried the same thing with iron nanomaterial. We got similar results for that one. So we sequenced them again, And fortunately, Bob had a sequencing unit here. So along with a sequencing unit. So we sequenced them in, but there are so much data. Bob is a genetic person, but he also doesn't know what to do with this data. Then what we did, we had a faculty member in industrial engineering who works with networking, Gehrts. So we worked with Gehrts to decipher what exactly is happening, So we tried to look details into this one, still very fuzzy. We thought we'll look a little bit into deeper one, still fuzzy. So we look still deeper, still fuzzy. Then I'm a civil engineer. I'm also trying to still look for a civil engineering type of problem because that helps me out. That's put me in the comfort zone. I'm not still finding a civil engineering problem here. Now we go there. Now here comes the civil engineering problem because once we, kind of rule out a lot of possibilities, find out the pathways. These are the pathways for gene signal transformation. So what we are looking for this hub here. This is like a traffic coming from here, traffic coming from here, and traffic coming from here. Now, if we do one thing, if we take this gene out, that means the traffic signal out, there should be chaos. There should be chaos, or there should be no flow of traffic for that matter, right? So this will give us a clue exactly if we can identify the particular pathway, which pathway is affecting our defense mechanism. I think that will help us identify the pathway, and then we can control that. That's the idea. So gene knockout, I didn't do it. Bob did it using a virus. He was a virus. He did it. He's very good in that. So he did it. Now what we have, we can now predict. We now know why exactly the whole thing happened. Why we held this, disease virulence. Because a lot of disease has happened because of nanomaterial and pathogens together. We can now know that. So what happened in the process? When we introduce nanomaterial, when you have a plant, when nectarotrophic bacteria attacks that plant, then what plant do? Plant typically will release a lot of jasmonic acid and less of salicylic acid. That's a plant's defense mechanism. So with a lot of jasmonic acid, the nectarotrophs will not be able to attack that plant. Okay. Now, with introduce of nanomaterial, what happened? The opposite happened. So what we see is a lot of salicylic acid, less of jasmonic acid. So we basically messed up the whole system, their system. Now our challenge is to design a new nanomaterial such that we can still apply zinc fertilizer in plants. So this is what we have found. Now this is a very good learning process. We are still working on some of the finer details of that one. Project is still on till end of summer. So we should be able to find out more details on that one. Now, we have learned a lot of things in this process. Now, what can we apply? Can we apply to something, I love so dearly? Can I apply to rice? Kind of a thing. That's what I was thinking. So what we did is we thought we'll look into rice because rice got a problem. Rice got a problem in a sense. Rice uptakes a lot of arsenic. Rice got a lot of arsenic. I'm pretty sure a lot of you love rice, right? A lot of you love rice, right? When you go to a Chinese restaurant, you like it, you like have rice, fried rice, right? If you're eating today, good luck with your rice. So, okay, anyway, so rice contains a lot of arsenic. And arsenic, as you know, is a poison of soils, right? Agatha Christie's, detective novel, there's a poison of soils, So it is very, very toxic, very, very toxic. It's very, very toxic. And we have two varieties of arsenic. One is the inorganic variety and an organic variety. Inorganic variety is the most toxic one. Organic variety is not toxic one. If we can convert inorganic variety to organic one, possibly we will not have a lot of effect. Within the organic variety, we have arsenic 3 and arsenic 5. So this is your arsenic 3 and arsenic 5. Arsenic 3 is even more toxic. Where do they come from? They come from a lot of geogenic, a lot of, even, volcanoes. Also, there are, these are basically regular minerals in the earth. Is there. Then also it comes from coal, oil refineries, mining, and everything. So whenever I teach 370 environmental engineering class, I always talk about this coal-fired power plants. So a lot of arsenic actually comes out in there. So now, so these are the problems we have. It can cause cancer. This is a major one. A lot of organ failures. Also, people do not know it can cause diabetes as well as heart problem. And arsenic can be found in a lot of our food as well. So if you look at arsenic, no part of the world is free of arsenic. Arsenic is there everywhere. So it doesn't matter you are a developed country or a developing country. So now, so if you look at, these are two pictures from two parts of the world. So this is US. So as you can see, North Dakota has quite a number of spots here, and Minnesota is full of arsenic. Anybody from Minnesota? Okay. Okay. Now, Minnesota is full of arsenic because of the glacial tilt. Because when the glacial tilt was deposited, that contained a lot of arsenic in there. So in fact, we visited a place called Winger, only 30 miles from here. Winger got an arsenic concentration in drinking water 12 microgram per liter. 10 microgram is the limit. So 12 microgram per liter is a town of 221 official population 221, and they had to spend $3.5 million to get the arsenic out of their drinking water, and that was a pain for the whole community, definitely. So that's an interesting part. A lot of constraint. In fact, even in the U.S., people are drinking water without treating the water. So then if you go to India, this is my home state, and this is whole Ganga and Brahmaputra Valley. This has a lot of arsenic in there. All the red spots are real arsenic concentration. Okay, so arsenic was first found in Bangladesh, thanks to civil engineers. Now, what we did is we decided to give them, hand pumps for irrigation of their water. As they irrigated, water table went down and arsenic started coming out, get oxidized started coming out. So we found from Bangladesh that arsenic is a problem for all of us, So now you go there, these are news items from Minnesota and North Dakota. And the most interesting part is the bottom part here. Arsenic more than the limit is there in our wells, in our wells that we use in our house. Almost 5% people have that arsenic. Why? Because many times we do not want government to come into our house, test our water. And we do not try to test it. We think our water is the best and groundwater is the best. It's always wise to test the water. You may not have it, but it's always wise to test the water. So this is a major concern for US also. This is also it involves a lot of economic burden on the community as well. So if you have, this is the initial signs of arsenic. It can, mostly skin cancer is very common. And other interesting part, as you can see, in India, 19% of Indian drinking water is there with lethal dose of arsenic. Very interesting. No, such a huge, one of the populous countries in the world, if you have 19% of their wells infected, you have a lot of people suffering from cancer, definitely. So now what we did in this work, so we got an NSF grant to work on rice. So it is a combination between US, Guinea, and India. Guinea also produces a lot of rice, and their rice also have arsenic. So Guinea was a challenge for us because Guinea speaks only French. So my student, one of my students, Seydou, was from Guinea, and he came as a Fulbright scholar. And he was our person of connecting us together. And we have started working on that one. So couple of years back, we had a scholar from India came on a Fulbright fellowship. So he worked on this particular problem. So what we did is, so we looked into various arsenic uptake paths and everything. So we thought we will try out some of the materials that we have developed. So problem here with arsenic in agricultural field, it is in a very low concentration. So if it's a low concentration, any reaction that takes place is, dc/dt is directly proportional to concentration. So if the concentration is low, your reaction rate is low. So it's very difficult to remove low concentration one, So now, so we thought we'll try out some of the nanomaterial. If we can concentrate the, concentrate the arsenic from soil, and we can easily remediate them because we can have microbial population who can possibly convert this arsenic to an organic arsenic. So that was our idea, because we want to take help of microbes in the process, but microbes will not work in a low concentration, area. So we started working on that one. So materials we wanted to use is a graphene-based material, which is decorated with iron. This is one of the work Tonoy has done. So this is a very interesting work. Ben was asking me when he came to the presentation, will be talking something about my area. Yes, this is an area, Ben, where actually this graphene-based material are good electron storage devices, and they're a lot of used in capacitors, actually, very much. Yeah, I have given your part. So yeah, okay. Now, so they work very well, and we found out the mechanism. We now know we are trying to find out alternative. Now, graphene is not necessarily a desired material, so we're now using biochars as an alternative to graphene. Okay, so we are also looking into some of those nanomaterials. Also, we are trying to use another nanomaterial called sulfonated nanomaterial, which basically we coat with a kind of a sulfur coating on top of that one. So what we did, we used those material, and we have grown plants out there, rice in our. So we collaborated with Upinder here, Upinder Gill's group. So we grown rice and we sequenced them after that. So what we got here, very interesting. We are yet to analyze all the data. So what we got is very interesting. What we got is, this is the control data. That means only arsenic being introduced to the rice plants. And next one, in 2.1, you can see those are the one where we have introduced nanomaterial with arsenic, such that nanomaterial can absorb the arsenic and help alleviate prevent plant uptake. So what we have found is, if you see the control and the nanomaterial being used, gene upregulation and downregulations are actually lower. That means plants are really accepting the nanomaterial as a friendly material. That's what it tells us. So we're yet to analyze the whole set of data. So basically, now, so we are working on that. I think we are hoping that will be really, we submitted a proposal to NIFA 5 years back. They rejected the proposal. They said arsenic is not a priority for us anymore, at that time. But arsenic is a priority right now. They are actually funding a proposal at this time. So now, of course, this is what research is. Of course, if you want to continue doing research, you need the next generation of researchers. So we have been trying to, of course, our graduate students are there. We also try to promote nanotechnology among school children. This is a school group from West Fargo. So they actually work on a biochar. They actually won the national award for e-cyber mission. They use nanobiochar, and they use it to remove phosphorus. So this is again what we did during Fulbright program. I went to a number of villages. These are village school where you talked about arsenic and also how we didn't talk details about nanomaterial, but we still talked about science in there. We are hoping that some of the students will come up and be the future researchers. These are our, current students who are really doing the job. And I took the, kind of privilege of presenting their work today. And these are, of course, some of the previous students we had. Of course, we had some of the super reactive groups. These are my collaborators from, there are more collaborators, but I only listing those collaborators who are involved in the presentation I made today. So there are collaborators from world over. We have collaborators from US, Brazil, then Guinea and India. So these are really, really working out very, very well. So with that, I like to, I studied in high school. I took Sanskrit. In Sanskrit, my teacher always used to say, now I'll do it in Sanskrit first, then I'll translate it for you. Keurani na bhushanti purushan, hara na chandujjala, na snanang, na bilepanang, na kushumang, na salankrita murdhaja, bani eka samalang karuti purusang, ya sangaskrita dhayate, kriyante kolabhushanani satatam, bhagabhushanam bhushanam. What does it mean? That means doesn't matter how nicely you are dressed, how nicely do your hair. It is the final content of your presentation, communication part, that is the most important. Don't worry about how well dressed you are, but make the presentation perfect. I take a special responsibility today. I took this presentation very seriously because I'm not presenting my work. I'm presenting my students' work and my collaborators' work. I hope I didn't disappoint my collaborators and students. But as you said, that it doesn't matter how well dressed you are, how nicely do your hair. It doesn't matter. I didn't take any chance. I thought I'll wear a new shirt today, and I also did my hair. In case you didn't like my presentation, make sure you compliment me on my, how I wear today. Okay, and these are different agencies that has funded us over the years. We are really grateful to be, getting competitive grants. Also thanks to the department and the college and the university for supporting us. And again, thank you all. It's really a pleasure to have you all here. Thank you.
Okay, well, now we're going to take time for questions. I'm going to ask someone on this side, so maybe Scott can, or the provost can. And I will walk over to that side. And so we will, if you have a question, we will hand you the microphone so we can get that recorded. So.
Okay.
Hey, Doc. I was wondering where you sourced some of the materials from for the original beads.
Say it again.
Where did you source the materials for the original beads?
You are talking
Like
about
the-
chitosan?
Yeah.
Okay. Yeah, chitosan is very easily available. So we get it from the, market. But chitosan is an interesting material because it is not like you buy sodium chloride, you get the same consistency, right? Same quality. But chitosan not necessarily same quality. Its base is different. So we normally buy in little bit bulk such that we get consistent result for all our experiments. That's how you do it. It's just easily available in the market. Yeah.
Wonderful presentation. So my question pertains to the first part
Okay.
of the nanoparticle of the phosphorus.
Yeah.
Have you done any testing on various soil types and how that reacts?
Okay. Yeah. I think that's a very interesting question. We have not done work yet. And that is one of the motivation for my presenting today, because, because I'm not an expert in the soil part of it, right? So if we can collaborate with somebody from soil science, I think it'll be good. We know a little bit about it. We are doing some work right now on soil part of how what kind of, bonds take place and everything. But I think that's an interesting area to look into, definitely. Yeah. Yeah. People have looked into that one because soil typically will be absorbing most of the time so it becomes legacy phosphate. I think that's a problem, right? So this is another interesting area. Can you release the legacy phosphate itself? You possibly don't need phosphate to work on phosphate you can just, if you can release the legacy phosphate for quite some time, that will possibly last for under 100 years, That's an interesting area, definitely. I'm sure you're pointing towards that. Yeah, thank you.
I have two questions. The first one is that how did you maintain control of the nano part of the material that you used?
Okay, yeah. So this is actually a very, very important question, because depending upon what nanomaterial you use, sometimes having a control may be very difficult. Take, for example, iron nanomaterial is very easy. You can create a bulk of yourself. You can use filings, iron filings as a control. But take, for example, zinc. In zinc, we really didn't have a control of actually micro, because we bought bulk zinc material and we analyzed the particle size. It still contains 30% of nanomaterial. That was a big challenge. So it is a little bit challenging. But in most cases, though, in case of iron or in case of, maybe if you're using, say, zinc, not zinc, sorry, manganese or something, maintaining, having microparticle is very easy, and they are very consistent in size, typically. Yes, some of them is difficult. You have to be careful. That's why many a time, just believing the commercial things that this is bulk, you should not believe that. This is not true. You have to test it yourself. Yeah. Okay, go ahead.
The second question is that you mentioned a lot of arsenic problems in Minnesota. In North Dakota, I think Lidgerwood on the southeastern part of North Dakota has problems.
Yes.
But
Yeah.
I don't know how seriously the state is looking at it, or have you looked at that?
No, actually, this North Dakota problem and Minnesota problem are two different problems. Minnesota problem is geogenic in nature, and North Dakota problem is anthropogenic. Because what we did is, in 60s, we used a lot of insecticide laden with arsenic to control insects at that point. And that area become, surgeon country become basically polluted with arsenic. So they cannot drink their own water. Actually, water is being supplied. This is super fun side. It is because it's soil contamination. It's very difficult to be cleaned up. This is one of the things we are doing. If we can really go the microbial route, possibly that will be a way to do it. So that is one of the things we are exploring right now. We are actually putting together a group to look on the microbial side of it. So that will be interesting.
Once when they were wondering of bringing water from Garrison diversion for irrigation purposes, the Oaks area, they had a lot of testing done on the groundwater wells.
Okay.
And I think arsenic was a serious problem. with that.
Sure. Yeah, I think it has to be. No, and North Dakota, of course, has uranium also. Uranium issue as well. Yeah. So that's other problem.
The southwestern part.
Yes. Southwestern
Uranium,
part. Yes.
yeah.
Yeah. Thank you. Yeah, I should have mentioned that. Thank you for educating everybody. That. Thank you.
Thank you. This was fascinating. It's great work. I'm curious, and it's getting ahead of it, but chitosan is forms chelates really easily. I mean, I barely understand this material, but that would mean that it likes metal ions, and it might be good for disrupting gram-negative bacteria. Is this stuff theoretically still present after if you've got your spinach and you're eating it? Are there microbiome disruption concerns if you?
So, okay, I'll answer the question in multiple parts. So depending upon what nanomaterial you use, like chitosan, one of the things we do is we would do biodegradation studies for all the material we develop. It has to be biodegradable and at the same time, not biodegradable fast enough. So it has to remain for certain time. There's no evidence of biopolymer getting into plants. Now, in case of nanomaterial, though, nanomaterial gets into plants, not necessarily in the form of nanomaterial. Take, for example, zinc will get into spinach, and it will create zinc phytate. Zinc phytate, it is not nanomaterial anymore. Zinc basically becomes ionized, and it uptaken by plants, and then plants sees it as an enemy, so they will, basically, create zinc phytate. So that's what happens. And there are evidences out there where people have seen nanomaterial getting into, plants, but those studies are done with a very, very high concentration of nanomaterial. So because they want to simulate very extreme conditions. In realistic environmental conditions, it doesn't happen. So in fact, I'm the president for Sustainable Nanotechnology Organization, which is a Washington-based organization. So our stand right now is that nanotechnology is very safe for agriculture, for any other purpose. It doesn't cause any harm. So I don't know whether I answered your question, but this is where we stand. Yeah. Okay.
I have a question from a remote participant, Daniel Erickson. He says, congratulations and great presentation. Even for a structural engineer, it makes sense to me. My question is, I know iron is good for females. However, iron may be detrimental to males. How do you defend that?
I do not know if it's detrimental to men because I don't know. It causes constipation, of course, if that is a detrimental part, So, my multivitamin, I do not take iron in there because, I have issues with that. But I don't think iron is detrimental. Everybody has anemia. It is irrespective of male or female. You can have anemia. I don't think iron harms. I don't think it's going to harm iron, I don't know if you educate me a little bit more, maybe I'll be educated at that point. I do not know really, male is detrimental for male,
I have a question that I can. So with respect to when you were doing your arsenic, did you see or do you predict that you would also see an entrapment of the selenium, which is also something that could help counteract the effect of the arsenic?
Sure. I think selenium, for that matter, silicon also are very interesting products, So we have not done that work, but there are reports about selenium being present and kind of a helping in the process, So as I said, I didn't do the work so on that one, but I have read report where selenium has been beneficial in the process. So, but silicon, just to give you an example, many times solutions really cause trouble. Like take, for example, silicon, for that matter. People use silicon to prevent uptake of arsenic. It worked perfectly. What the plant did with the silicon uptake pathway, they took the silicon and kind of not take the arsenic. So now problem was there's no arsenic in the rice, but the problem was the fodder, because fodder has to be taken by animals. So they have problem with their teeth now because there are silicon out there. So it is a kind of you have to really look into you cannot solve problem like a DDT guy did, So we have to go beyond the DDT thing. You have to look at the whole cycle, But selenium is so far good. There's no adverse effect on selenium as such. Yeah.
Hello, Achintya. Congratulations and nice talk. As an environmental engineer, I want to ask you some question related to that field.
Okay,
And recently, I read a paper in EST about there's a concern between the new people, environmental researchers are trying to see the new contaminants and try to control them. And researchers are trying to find one more, another one. But a lot of common contaminants are being neglected by funding agencies, researchers, for example, arsenic. We have a serious problem there, but researchers, not a lot of researchers are working on it. So I'm thinking that in the future, probably we can solve the new problem. At the same time, we can also solve those old problem or existing problems. So I'm thinking what is your opinion about this future research in terms of solving both problems at the same time, and how can nanotechnology be used to solve all those problems?
Sure.
Thank you.
Sure. No, thank you. I think, yeah, you're right. So take, for example, PFAS. We all of us doing PFAS right now, right? It's just a new thing. We have to always jump into there. The old thing get kind of neglected many times. Arsenic has been neglected enough. Fluoride has been neglected enough. So we do not do enough work. You know why? Because it doesn't affect us, the middle class. The middle class people are not affected by those contaminants. The poor, underprivileged people are affected most. That is one of the reasons. This is the equity part is missing from there. Now, if you look at, nanotechnology being used for remediation of this one, so it is very interesting because arsenic, when you remove arsenic is a non-destructive chemical. So you can actually phase transfer arsenic from liquid phase to solid phase. You cannot destroy it. So that's the problem. Arsenic will be there somewhere. Now, what to do with the arsenic then? So one of the things, arsenic actually used extensively being used in electronics. You need arsenic in electronics. Then also arsenic is used for cancer treatment. So nano arsenic is big for cancer treatment. That can be a circularity coming out there. I think we need to think, like I gave you an example, 5 years back, we had such a nice proposal with Jason White from Connecticut Experimental Station, and all of us wrote a proposal. We got rejected because they said arsenic is not a priority for us. But now they are seeing it a priority. I think it will come back, because earlier, baby food didn't have any advisory for arsenic. Now there's an advisory for baby food, rice-based arsenic, the rice-based baby food, because you cannot have more than 100 microgram per kg of baby food, in arsenic per kg of baby food, like rice-based. And they also advise that babies should not be given only rice-based, thing. So there are emphasis coming back. I think it's a question of, how our politicians think and how NSF and USDA thinks. I say many times, right? I suppose our objective is we need to also give input to those agencies such that they can also, they live on inputs, right? They live on inputs. So we should be possibly doing that. I see that coming back again, some of the areas, yeah. And we should not be really kind of a disheartened, so it will come back, so yeah.
We have one last question on the Zoomer.
I have a question from remote, and I guess I should clarify that Daniel Erickson is just conveying questions from others. But anyway, this question says, zinc is the most important micronutrient in human health. However, estimates from the U.S. Geological Service indicate that the global zinc resource will be depleted in about 20 years. What are your thoughts about how we might stop using zinc in non-food applications in order to preserve it?
This is beyond my expertise. I do not know. I do not know what exactly. I know there are alternative materials being used. Like take, for example, because of dependence or our dependence on art metal on a particular country, and we have been really doing excellent research nowadays. We are finding out alternative to art metal. So I'm very sure that, I know that my program manager from NSF, also program manager from USDA joining today, they are listening. So maybe we should talk to them, so about the zinc problem as well. So I think I do not know really. I don't have an answer for that one. I'm sorry. So yeah.
Okay, well, thank you very much for joining us today, and I'll ask the provost to give the final words.
Please come up here. Thank you very much. It was a wonderful lecture, so thank you. I'm NDSU Provost David Bertolini. President Cook sends his apologies. He couldn't be here today. He's preparing for a governor's visit with the OMB office tomorrow morning, so he's cramming, I think is what we would say in that. It's important to note that the faculty lectureship is incredibly important in NDSU. It represents the best that the university can offer. And it's also important to note that it's a rigorous process that means that you have to have an exemplary portfolio of research and teaching, and you're also judged by your peers. And to be selected, in this case for the 63rd lecture, is a remarkable achievement. So we send our congratulations to you. The other goal of the lectureship is to also provide or have a mechanism to bring very complex, maybe very specific expertise to both your peers, but also to novices or uninformed people like myself. So it's a way to share it among ourselves. And I guess two takeaways I have is to start worrying about arsenic, and also to know that Popeye is a metric in high level research. So that's really, really important. I didn't know that. But you did a wonderful job.
Thank you.
The other thing to note too is that a land grant mission is unique in the sense that we have to do two things that are incredibly difficult, and it puts incredible demands on our faculty. One is to have high level research, right? To work at the highest level possible in the nation and the region, and also make that available to the state and to the world that makes a difference. The other part is that we have to be excellent teachers, that the land grant mission is to educate the next level citizens of North Dakota and the region to do well in the world and make a difference. And Dr. Bezbaruah has demonstrated that on all levels. And in fact, I'll go out on a level that, at least on the teaching side, I think in the majority of conversations, within the first or second thing that he's ever said to me is, my students did this, or my students are that. And it's just truly remarkable. So I'm grateful that you're here, and I thank you. And I'm going to present you this little plaque of today's episode. So thank you so much.
Thank you.
Are you going to close out? There's also a little-
Okay, thank you again very much for joining us today. If you would join us and the good doctor here in the Butte Lounge, I did see cake and some drinks. And so thank you again, and please bring your questions up front for Achintya and the rest of it. So thank you.